So the sex determination region, it turns out it's a region on chromosome 2 that's been mapped before. We knew from the Pinot Noir reference what that's supposed to look like. Chardonnay, we actually sequenced it really well, so we had 2 hermaphroditic haplotypes. Pinot Noir, we think the reference genome is a female haplotype. We thought and hoped that what we could do is take the sex determination region and find out how the sex system was broken when it was cultivated. So we did all this mapping of all these genes. We found out some genes that differed between the hermaphrodites and the females, but in the end, we couldn't figure out what happened and why the sex determination region broke. And I was super, super upset, but life's okay. We were hoping we'd find a smoking gun that tells us what happened, but we didn't find it. We also think that actually the sex region was broken a couple of times, and that's part of the problem. So we're still studying that. But I do want to tell you about this, probably the most studied phenotype in the history of plant biology, though people would disagree with me. This is the phenotype I showed you before. Dark grapes, green grapes, transposable element, structural variation, turns off the gene. So you need to have the structural variant to get green grapes. But you also need another gene to have a null mutation. So there are 2 of these MIB genes right next to each other. Remarkably and very interestingly, these 2 MIB genes right next to each other, they have null mutations and they're heterozygous in many dark buried lines. What that means is there's one set of copy functional MIB1 and MIB2, one set of not functional MIB1 and MIB2. Why it's heterozygous, who knows? So then what has to happen if you want to make green grapes from purple grapes is those null mutations either have to become homozygous or hemizygous, right? You have to get rid of the functional copies. Well, when we were looking at our new Chardonnay genome, it's the only green grape that's been sequenced so far, we found this really big inversion relative to Pinot Noir and also to Cabernet Sauvignon. This big region, this big inversion, by the way, includes those MIB genes that change the color of the grapes. Okay, so wait, this is a complex story. You need 2 genes to be knocked out, and now we have an inversion of the region. We looked into the literature, and it turns out that someone else has studied this a little bit. This is Tempranillo grape, my favorite variety. There was a somatic mutation to green grapes. And it turns out when they looked at coverage statistics, they found this region of hemizygosity in chromosome 2, or exactly where the berry color locus is. So somehow, during the somatic mutation, the functional MIB1 and MIB2 were removed from the genome. They thought that it might involve an inversion. And in fact, they had some evidence to suggest that's the case. So now we have 2 cases of green grapes showing up, including inversions. And in fact, we made a phylogeny of a bunch of grapes, and we can show, based on this phylogeny, that green grapes originated bunches of times, right? So this isn't something that happened once. We had purple grapes and then different varieties of green grapes. We had independent origin of green grapes. So we were curious. It's known you have to have those two knockout alleles of MIB1 and MIB2, but do you always have an inversion? Briefly, we looked at all these using coverage statistics, and this region here shows you where the color locus is. It's hemizygous, so in this combination of a green grape and a purple grape, the green grape derived from the purple grape, you again see this hemizygosity. And when we really look carefully, there's evidence that in every case there's a green grape, there was in fact an inversion. But it's always in the same region, but slightly different size. So in these 8 cases we looked at, or actually we looked at 6, the inversion's either around 4 or almost 5 megabases and includes 134 to 176 genes. So these inversions, we think, are somehow making the region hemizygous, and that's how you get a green grape. I think that's interesting for a couple of reasons. One, why do you get an inversion in the same region so often? We don't know. There's some studies in sticklebacks in humans that suggest some regions of genomes are more fragile than others. That could be the case. Or it could simply be that people see green grapes and select. Maybe these inversions happen all the time, and usually you don't see a phenotype. We just don't know. We also tested whether all these 118 genes are in any way related to grape color. Maybe, you flip the region around and it changes the expression of genes. We didn't see any evidence for that. So we think these inversions are actually causing the mutations that make this region hemizygous, and then you have the two knockouts. Okay, some of your grapes, sorry, I've gone too long. Grapes experience little genetic bottleneck. Clonal propagation provides an opportunity to hide recessive deleterious variants. So through time, I suspect this is true of all clonal plants, you're accumulating deleterious both SNPs and SVs. 1 in 7 genes is hemizygous. I was shocked by that. Maybe it's not so shocking. The sex determination is a hotspot for SVs, but I can't tell you what happened genetically to break the sex system. Independent inversions, and I think this is pretty novel, have helped drive convergent berry color in evolution. Again, berry color evolved multiple times into